Dear students, welcome to Bioratna. I am Chandana Sengupta and today we are going to discuss the topic Replication of DNA from the chapter Molecular Basis of Inheritance. Replication of DNA. Students, it was in 1953 that Watson and Crick, they gave the double helical structure of DNA, the famous double helical structure of DNA. But it was only after 5 years in 1958 that Mendelssohn and Stahl, they gave the proof that DNA replication is of semi-conservative type. It is semi-conservative mode of replication. What is replication? Replication means to form a replica of itself, to form a copy of itself. And when Mendelssohn and Stahl gave the proof that it is semi-conservative type, semi-conservative means from one parental DNA strand, 2 daughter DNAs are formed and each daughter DNA is having one of the strands of parental DNA and one new strand. That is called semi-conservative. Each one is conserving half of the parental strand. So the proof was given in 1958. But in 1953 itself, when Watson and Crick gave the structure of DNA, at that time they hypothesized, they proposed, and they could see that DNA replication is of semi-conservative type. How they could see? Let us see the structure given by them. This was the double helical structure. Now, let us make this one straight so that we can understand this properly. This is one of the strands which is having, suppose, the nitrogen bases. You can write A, C, G and G. Okay. Then the other strand, this other strand, it will have the nitrogen bases T, T, G, C, C. This is complementarity rule which you know already, and you know that how many hydrogen bonds are present between A and T and between C and G. Now looking at this diagram, Watson and Crick, they could see that these are the 2 strands and these hydrogen bonds by which they are held together, these are very weak bonds. If these hydrogen bonds are broken down, then what will happen? Then these 2 strands, they will separate. So, when these 2 strands, they separate, this strand, I am writing this side, this is its 5' end, this is its 3' end. And this another strand, its 3' and 5' end. This is having T, T, G, C, C, G, C and C. This one is having A, C, G and G. Now both of them will form the new strand. When they form the new strand in this 5-3 direction, then see what happens. This is A, C, G and G. This one, this is T, T, G, C and C. So, A, C, G, G, A, C, G, G, T, T, G, C, C, T, T, G, C, C. Same here. A parental strand which on which the new strand is formed, it is called template strand. This is the template strand, and this is the newly formed strand. This is the new strand. And you can this type of replication where the two daughter DNA, each one is having one of the strand of parental one. This is called semi-conservative type of replication. Now let us see the types of enzymes which are involved in replication of DNA. So we are studying the enzymes which help in the replication of DNA. The most important of all enzymes is DNA dependent DNA polymerase. DNA dependent DNA polymerase. Why is it called so? See, here it is written DNA polymerase. It is easy to understand. It is doing the work of polymerizing the nucleotides of DNA. That's why it is called polymerase. Now, for that polymerization to form the new strand, it needs the help of one of the strand of DNA. This is the template strand. Just you have seen that on one of the old strand of DNA, the new strand is formed. So this template strand, with its help, the DNA polymerase, it then forms the new strand. That's why it is called DNA dependent DNA polymerase. This enzyme, it is very fast and it is very accurate. It is very accurate. Rarely it does any mistakes. It is very fast. How fast it is? You will get the idea that in E. coli, the number of nitrogen bases, they are 4.6 into 10 to the power 9. And the whole replication is completed in 38 minutes. That means just in a single second, in 1 second, 2000 base pairs are polymerized. 2000 base pairs just in a second. That fast it is. But as it is fast, it is not that it makes many mistakes. Why many? It rarely does any mistakes. That mistakes also it edits and corrects it. That it is very accurate, because if it is not accurate, if there is any change in any nitrogen base, then it will give rise to mutation. So it is very accurate also. So this is one enzyme. The second enzyme is helicase. What is helicase? Helicase helps in breaking the hydrogen bonds between the two DNA strands. The DNA strands are helically wounded around each other. Now this enzyme goes on breaking the hydrogen bonds, so the DNA molecule unwinds and it separates as two single-stranded DNA. So the function of helicase is to unwind the two strands of DNA. Two strands of DNA. Next, topoisomerase. Topoisomerase. What does this enzyme do? See, if in your house itself you would have seen that if the two threads are wounded around each other and you hold the two threads and try to pull it opposite to each other, try to separate these two strands, then the lower part it coils. It immediately coils. A tension occurs and it coils. Same thing happens with DNA strand. When the helicase is unwinding the DNA, then the lower part of DNA, it becomes coiled. Then topoisomerase, it makes one cut in one of the strands. The tension is immediately relieved and then it seals it. So the function of topoisomerase is to relieve the tension in two DNA strands which had occurred due to unbinding. Other than these enzymes, there are other enzymes also like ligase. Its function I will tell you when we come across the step which it catalyzes. Next we see the substrate for this replication of DNA. You will say that it is easy. It is the polymerization of nucleotides. So substrate is nucleotide. This enzyme DNA dependent DNA polymerase is polymerizing the nucleotides. So the substrates are nucleotides, but it is not so. We will see what is the molecule and how it acts as substrate also. At the same time, it provides energy too. This process is energetically very expensive. It needs lot of energy, and every nucleotide, it takes care of its own energy requirement. Let us see the substrate for DNA replication. The substrate for DNA replication, it is nucleoside triphosphate. Let me first draw one nucleotide, and then we will see what is a nucleoside triphosphate and why is it named so. This is a deoxyribose sugar. This sugar, when it is attached to a nitrogen base at C1 position, then it is called a nucleoside. Then when it is attached to the phosphate group at the C5 position, then it is called a nucleotide. So this is our complete nucleotide. Now, two more phosphate groups are attached to it, and these are two very rich energy bonds. Now, if we write these three phosphates together, then the remaining molecule, this is one sugar and one nitrogen base, that is nucleoside. That's why it is nucleoside, and the three phosphates, so it is triphosphate. This is nucleoside triphosphate. Very important question for MCQ as well as in your board question. Now see, these two bonds, they are very energy rich, and when these two bonds are broken, then it releases lots of energy, and this energy is utilized in the formation of the new DNA strand. This was about the substrate which is carrying with it its own stock of energy. Now let us start with the process of replication. Students, the DNA molecule is a very long molecule. It only opens up at a particular small place, and this gives rise to a fork-like structure called Y fork. This is called Y fork. So one is 5 prime 3 prime, this is suppose 5 prime 3 prime, and these lines, they show the base pairs. So these are the base pairs. Fine. Now this is the replication fork. This is replication fork, or also called Y fork. This is also called Y fork. Now the DNA replication will start, and there is one problem. What is that? The new strand, the DNA dependent DNA polymerase, it can do this polymerization only in 5 prime 3 prime direction. So on this template strand, it has no problem. It has got its 5 prime end, and it has started. But look here, another strand, it will have its 3 prime end this side. So it cannot proceed this way. So what solution it finds, it does not replicate in this direction, rather it replicates in this direction, because here it is 5, here it is 3. But what happens due to that? It has opened this much. We will see how this strand becomes a continuous strand without any breaks, and this strand will have many breaks. See, now this strand, it was opened up to this much. Now I will open up it up to this. So this much we have drawn, and then it has opened up. This strand is 5, 3, and this is 5, 3, 5. Okay. Now this strand, it will form a continuous strand, but this strand, at first it has formed only this much, a short one. Now again it will form another short segment. So this is discontinuous. This is discontinuous strands, whereas this strands, it is the continuous strand. This is the continuous strand. You understood the problem why this happens, because it has to take the 5', 3' direction. So every time this one is opening up, then it is forming a small segment. Now previously I had told you about the enzyme ligase. This enzyme ligase, जोड़ने वाला enzyme. Now this ligase, it joins all the fragments of discontinuous strand. This is how DNA gets replicated. The DNA polymerase, which is moving in one direction, once the replication is completed, it moves in opposite direction and it edits its work. It sees that whether any mistake is done, and then that base is replaced. One more thing you need to know is ORI, or origin of replication. What is origin of replication? In DNA, it is not that the replication can start at any point. There is a fixed point from where the origin from where the replication starts. So it is called origin of replication. So this is what one interesting story of replication of DNA, the semi-conservative replication of DNA, how the one parental strand, it separates into 2 and they act as template strands. Then the DNA dependent DNA polymerase, it forms 2 new strands on the old strand. I hope you have understood the topic properly. If so, and if you are new to my channel, then please do subscribe, like and hit the bell icon so that you get all my notifications. Thank you for watching the video.